Hello everyone, I'm SHEENA and welcome to my channel Comprehend Biotech. Before we start, in case you are here by accident and you have no clue what the topic is today or what biotechnology is about, please do not leave so soon because I have something for you as well. If you check out the description of this video, there will be a timestamp that says stay motivated. Please click over there and you will have a motivational quote. So at the end of every video or every class session, I actually give out a motivational quote. So maybe that is just something that you needed to start your day strong or to even end it at a positive note. So please do check that and then you're free to leave. And for others who have come for the discussion today, welcome. Today's topic is cDNA-AFLP. So we'll be seeing what is cDNA-AFLP in as much detail as we can. So I have divided the procedure into a number of steps, put in the best diagrams that I could find online. So I take a lot of help from the old online resources that are there. So I hope you will be understanding the major steps involved in cDNA-AFLP and why it is used. And yes, let's get started. Welcome again. So the topic for discussion today is cDNA-AFLP. Let's start. So cDNA-AFLP is a technique that we will be majorly using for gene expression studies. But before that, we need to know what is AFLP. AFLP or Amplified Fragment Length Polymorphism. It is a PCR-based technique which uses selective amplification of a subset of digestive DNA fragments to generate and compare unique fingerprints for genomes of interest. So we'll be seeing the steps in detail, so don't worry about that. Basically, AFLP is the technique which we'll be following, which will be used on cDNA. Okay, so if you understand AFLP, that is the same thing. Only the first step is to get cDNA, right? So AFLP, why is it used? AFLP detects polymorphisms in DNA when no information about the genome is known. So a DNA polymorphism is a DNA sequence variation that is not associated with any observable phenotypic variation and can exist anywhere in the genome, not necessarily in a gene. So basically, we're going to check for or understand where the polymorphisms have occurred. Now, what are polymorphisms? They're observable phenotypic variations. Phenotypic means something that you can see in the physical nature of an organism. Okay. So now I'll be telling you what could be the types of polymorphisms. Okay, so polymorphisms can be single base or thousands of bases. So you can have a change. To give you a flashback, DNA is composed of nucleotide bases, right? And when you have your genetic material, there's an order in which these nucleotides are present. And for an organism, it has a certain order of nucleotides. If there's a change in one single base, that is called polymorphism. Okay, now this change could be there in a single nucleotide or even multiple thousands of nucleotides might have been, changed. Suppose there was an adenine in its place, a thymine might have come. So that is called single nucleotide polymorphism, a change in the single nucleotide. And you might have studied about sickle cell anemia, right? So sickle cell anemia is also caused by a single nucleotide polymorphism. Okay. So if the location of polymorphic sequence is known, it can serve as a landmark or marker. So landmark is something which gives you a distinguishable portion to identify a particular genomic segment. Okay. So amplified fragment length polymorphisms are differences in restriction fragment lengths caused by SNPs or indels. I'll be telling you what those are, that create or abolish restriction endonuclease recognition sites. So if you know about restriction enzymes, you might also know that the restriction enzymes, they go and cut a particular restriction site. They have a particular recognition site. So if you take an organism, if you take its genome, if you're cutting it with restriction enzyme, a particular restriction enzyme, you'll get a fingerprint. So every time you take that genome and you put the same restriction enzyme, the same pattern will be repeated. So that's why that's called a fingerprint, right? When there is an SNP or when there's a polymorphism, there will be change in the base, nucleotide base of the genome, right? Whichever organism you're considering. At that time, what will happen? If there's a change in the nucleotide, the recognition site is modified, right? So the next time when you add a restriction enzyme, that restriction enzyme will not be able to come and identify that particular site because there's a single nucleotide change that has occurred. Okay, so for example, E. coli has a 6 base motif. It will only recognize that particular motif and go and cut there. If there is a change or difference even in one nucleotide, it won't be able to recognize. Then what will happen, whatever pattern you will get of the fragments after restriction digestion, that will be different. So that's how you compare two organism, one with polymorphism, one without polymorphism. They will both have different fingerprints, okay, or DNA fingerprints. So what is SNP in a bit more detail? The most common type of polymorphism involves variation at a single base pair. So that is what is this single nucleotide polymorphism. Actually, it is like abbreviated as SNP, but you'll be hearing it as SNP. Okay, it is called as SNP. So called a single nucleotide polymorphism or SNP. Okay, scientists are studying how SNPs in the human genome correlate with disease, drug response, and other phenotypes. What is indel? We said that polymorphism could also be caused due to indels. What is indel? So insertion deletion. Okay, it's a short form for insertion deletion. So as the name suggests, you'll be able to understand what it is, right? So indels markers are an important class of molecular markers for mapping heritable traits in animals and plants because the markers with large insertions or deletions can be directly scored on agarose jars. It's nothing. Suppose a particular set of nucleotide has been inserted into the genome. You already had a genomic segment, and due to some mutations, due to some variations, a particular set of nucleotides has been inserted. That is called insertion. Suppose from another place, a particular set of nucleotides have been deleted. That is called deletion. And in case there are more than 10 base pairs that have been inserted or deleted, these can be easily detected by agarose gel electrophoresis. Okay. The patterns obtained from different strains are polymorphic due to, now why do we get these polymorphisms? One is mutations in the restriction sites. So of course, if there's a mutation in the restriction site, the restriction enzyme will not be able to come and cut at that particular point. So there will be, of course, you'll be able to observe the differences, right? One which is not mutated, one which is mutated, you'll be able to compare it easily. Mutations in the sequences adjacent to the restriction sites and complementary to the selective primer extensions. There are some restriction enzymes which do not cut exactly at the recognition site. What they do is they have a cutting site, recognition sites, for example, in the beginning, and their cutting site might be some 15 base pairs away from their recognition site. So such, recognition enzymes are also, restriction enzymes are also there. So mutations in the adjacent sequences can also affect them. Then insertions or deletions within the amplified fragments. We discussed that. So basically, I'm just trying to put it concisely, what causes polymorphisms and how does, what are we actually trying to study? What are we trying? So we saw what is AFLP and what are the reasons why there could be polymorphisms. Okay, when we are studying the differences, what could be giving rise to those differences. So now we'll go into what is cDNA-AFLP, and under this only we will be studying the steps involved in it. Okay, so basically it is the steps for AFLP, but one addition will be the formation of cDNA in the beginning. Okay. So cDNA-AFLP or cDNA Amplified Fragment Length Polymorphism is a comprehensive transcript profiling method that pinpoints genes differentially expressed between individuals and groups of phenotypes under different conditions. So here the basic concept is shifting to what you can say at the mRNA level or the expression level. How can I explain it to you? If you're talking about normal DNA fingerprinting, you're comparing the differences between two DNA samples. But here, it is the difference between which genes are expressed, right? So it gives you a transcript profiling. It's a transcript profiling method. What is transcript? If you remember the central dogma of molecular biology, you have DNA. DNA is transcribed to mRNA and mRNA is translated to protein. So when you have DNA transcribed to mRNA, mRNA is called a transcript. Okay, so in cDNA-AFLP method, we are actually going to get a profile or a fingerprint, or that is what is called a transcript profiling. Okay, profile for the transcript, that is the mRNA that we have, the expressed part. Okay. Now, why do we need to do this? Basically, when does gene expression change? If you look at your body, okay, you have different cells in your body. All the cells have the same DNA, right? But all the cells do not have the same functions, right? So if you look at the cellular level, one cell is acting as a nerve cell, one cell is acting as your blood cells, even though the genomic DNA is the same, right? So what is happening is, at particular stages of life, only particular set of genes present in your DNA are being expressed, right? And those genes will be different based on the different conditions. So, for example, if you want to study the gene expression difference between two plants, one plant is growing in particular condition, another plant, the same species of plant, you're growing in another condition. What will happen is, if suppose it is flourishing well or flowering well, that means some other kind of genes have been expressed in that plant. So you'll be doing the profiling of both, and you'll be finding that exactly what has changed. If you look at DNA, nothing has changed in the DNA. It's only the expressed genes that have changed, right? So that is where transcript profiling is important. So that is what is being said over here. So cDNA-AFLP pinpoints genes differentially expressed between individuals and groups of phenotypes under different conditions. So for this purpose, the standard AFLP procedure is performed on cDNA, that is the complement of mRNA generated from a given tissues. So I hope by now you know what is the complementary DNA. DNA is a normal genetic material that you have. But what we have found out, okay, from DNA we get mRNA, right? mRNA reverse transcribed into DNA. That DNA is called cDNA, okay, complementary DNA. So what is the difference between normal DNA and cDNA? The major difference is, when DNA is transcribed to mRNA, mRNA is modified, right? A lot of exons and introns are present. So those introns are removed and you have exons spliced together. So introns are those regions which do not code for any gene. So you have your DNA has coding regions and non-coding regions. Not everything are genes, right? So when mRNA is derived from DNA, it has exons, which are the ones which will code for a protein, and introns, separated by introns. So two exons might be separated by an intron. So removal of that intron and joining together of those exons. So that is called mRNA splicing. So after that we have that mRNA, when you reverse transcribe that, reverse transcribe means you're going to reconvert it back to DNA, right? That time what will happen? This cDNA will only have the exons in it. That means the expressed gene. Only those portions will be there in this DNA. So that is the difference between your cDNA and your normal DNA, right? So compared with other transcriptomic techniques, cDNA-AFLP is fast, it requires only a small quantity of mRNA and can be applied in the absence of prior sequence data. It is reproducible and it is a very sensitive method. Now let's start with the actual procedure of how cDNA-AFLP is done, okay? So many modified protocols you will find, but majority of them have these major steps. Okay, so what is the first step? Of course, as we know, we need cDNA, right? So first step is the reverse transcription of mRNA to cDNA. So whichever sample you're going to analyze, first of all, you have to collect the samples. You are going to isolate the RNA from it. Okay, and then you're going to carry out reverse transcription to get cDNA. So that is the first step. We're not going into the description of about reverse transcription because again that's a very basic thing that we need to know before we come to cDNA-AFLP, right? So the first step is getting your cDNA. The second step is digestion. So when you hear digestion itself, you know it will be the use of restriction endonucleases. Here we're going to use two types of restriction endonucleases. One is MS-even, which is a frequent cutter. Frequent cutter because it will be having recognition sites along the genome or along the cDNA, frequently. And it has a 4-base motif, that is, it recognizes 5'TTAA, and it's a very frequent sequence that will occur in your DNA sequence. Okay. Another restriction enzyme that we use is EcoR1, restriction endonuclease, which is a rare cutter. It's a rare cutter because it has a recognition site which has 6 base motifs. That means it recognizes GAATTC. So of course, such a sequence will occur at a rarer frequency than your frequent cutter's recognition site. So you basically consider using a rare cutter and a frequent cutter. Now these two are the commonly used enzymes in cDNA-AFLP, but you might change these according to your experimental needs. Okay. Now the basis for using two restriction enzymes. Why are we using two different types of restriction enzymes? The frequent cutter or the frequent restriction enzyme, that is MSE1, will produce small DNA fragments that will amplify well and are in the best size range for separation or denaturing gels. So basically how we are going to analyze this, we are going to carry out the gel electrophoresis. So while doing gel electrophoresis, you need DNA fragments, right? You have a whole cDNA, long cDNA molecule, you're going to cut it into different pieces and then run the gel, and the sizes will separate according to their, the pieces, DNA fragments will separate according to their size, right? So since you are using a frequent cutter, it will be cutting your cDNA at very frequent intervals. So you will have very small pieces of cDNA, which will be able to separate out very easily on gel electrophoresis, okay? Then the number of fragments to be amplified is decreased by using the rare cutter, and this limits the number of selective nucleotides desired for selective amplification. So I will be telling you what is selective amplification and how using this rare cutter, the number of fragments are reduced. Okay, to explain that, actually I had scribbled down a diagram. Let me see if I can explain this to you. Why do we need to reduce the number of samples that we'll be running in the gel electrophoresis? I have scribbled it down. It's not a technical diagram, but I hope you will understand. So for example, you're going to analyze two samples, sample 1 and sample 2. You're going to isolate mRNA from it. You're going to carry out reverse transcription, right? So this mRNA formation happens inside the cell, right? Then you isolate this mRNA, you do the reverse transcription, this happens outside, okay? You get cDNA. Now, if you are not using any kind of selective techniques or selective restriction enzymes, what will happen? We are going to use selective PCR over here, okay? You'll understand much there, but I'm trying to give you an answer to why do we need to, reduce the number of sizes, fragments. So you're just normally putting in your frequent cutter, and the frequent cutter is going to cut the cDNA at a lot of points. Okay, so you will have many small fragments. Okay, what will happen? That will crowd up your gel electrophoresis setup, right? It is going to crowd up. So how do you find the difference between 1 and 2? What are the genes expressed over here? What are the genes which are not expressed over here? How do you even find that out when this is so much crowded? So to avoid that, we'll reduce the number of fragments that we are going to analyze further. Okay, so that we do something called selective PCR. After that, when you do the gel electrophoresis, you will have a sample size which will help us to identify more. So you can see over here, this one is missing over here. This particular fragment is missing over here, and it's so well seen, right? For example, this particular piece is not over here. So it's easier to compare two samples when the number of fragments of cDNA that we are going to analyze has reduced. So that's why we are trying to use two restriction enzymes, and then further we are going to go for some pre-selective and selective PCR. Okay, so you have to avoid this particular crowding situation. So just taking the raw cDNA and doing restriction enzyme digestion and just putting it in the gel, it will give you a crowded one. You will not be able to, even though differences will be there, but it will not be analyzable by us. Okay, I hope that's a word. Okay, we will not be able to analyze it properly. Okay, and so that's why we'll be carrying out selective PCR, so that the number of fragments that we are going to analyze will reduce, and you'll be able to clearly see the difference between two samples. What has been expressed in the one which has not been expressed in the other. So the transcript profiling can be done in a much better way. Okay, so yeah, that is what is being said over here. So the use of two restriction enzyme makes it possible to label one strand of double-stranded PCR products that prevents the incidence of doublets on the gel due to different mobility of two strands of amplified fragment. Again, I'll explain it to you with the diagram, you'll understand it better. Using two different restriction enzymes gives the most agility in tuning the number of fragments. This is what I explained to you right now. A large number of different fingerprints can be created by the diverse combination of a small number of primers. So this is the first step over here. You're going to do the restriction digestion, right? So first of all, you can see this is your cDNA. You're going to add restriction enzymes, MSE1 and EcoR1, and also DNA ligase. And now why that is going to come? Because the second step is actually addition of adapter. I'll come to that, but just look at this particular step right now. cDNA plus the restriction enzymes. So MSE1 has a restriction site of AATT, right? 5-TTAA. And if it sees that, it will cut in between T and T over here. Same way in the opposite strand, 5-TTAA is here, so it will cut over here. EcoR1 on the other hand has a recognition site of GAATTC, and it always goes and cuts between G and A. Same way on the opposite strand, on the complementary strand, it will go and cut over here. That's it. That's the first step. Okay, restriction digestion. You have put MSE1 and you have put EcoR1. Now also remember that it's not necessary that at one end MSE1 will cut and at other end EcoR1 will cut. If you have a restriction site for MSE1 over here, MSE1 will cut at both these ends. Same way, there could be a restriction site for EcoR1 over here. This particular diagram is just an example of where MSE1 will come and cut and where EcoR1 will cut. There will be fragments which has both sides cut by MSE1 or both sides cut by EcoR1. Okay, so this is the first step. This is another diagram. So all through this presentation, you will see different diagrams. I just tried to assimilate as much as I could to explain it better. I hope you'll be able to understand also. This is another diagram that shows cDNA where EcoR1 is cutting over here. So it will be cutting between G and A over here, and MSE1 will be cutting between T and T over here. Okay. So digestion of genomic DNA to completion. Digestion results in large number of fragments such as MSE1 and MSE1. That means they are both cut by the same restriction enzymes, predominant and to be excluded from visualization. So later on, those fragments which have been cut at both ends by MSE1 will be excluded from our analysis. We'll see how that is done. Okay. You will also have pieces which have EcoR1 and on one side and MSE1 cut by MSE1 on the other side. This is the example for that one. And also another set of fragments which are cut at both ends by EcoR1. Okay, so this is the same thing. Okay, then what is the second step? So that is the digestion step. I hope that is clear. You're going to add a rare cutter and a frequent cutter, MSE1 and EcoR1. They will both go and cut at their respective positions. So you will have cDNA fragments. cDNA will be cut at many points. You will have cDNA fragments. Some of the cDNA fragments will be cut by MSE1 on both sides. Some will be cut at one end by EcoR1, one end by MSE1, and others will be cut by on both ends by EcoR1. Finally, those with MSE1 cut sides will be on both ends will be avoided. Okay, we'll go to that. Don't worry. So after restriction digestion, why did we do this? After this, we'll be doing the addition of adapter. So that is called the third step. Adapter ligation. So two different adapters, short double-stranded DNA with sticky ends. So adapters are nothing but they're also double-stranded DNA with sticky ends. Okay, those are ligated to the digested fragments. So if you look at these digested fragments, they also have a sticky end, right? So you have adapters which are also having sticky ends, and they will come and join over here with the help of the enzyme DNA ligase. So one adapter will complement to MSE1 cut end and the other will complement to EcoR1 cut end. Adapters turn sticky ends to blunt ends, modified the restriction site. So these adapters are designed by us. We know what will be the restriction we know what restriction enzymes we have used, so we know what the restriction sites are. According to that, we'll be designing our adapters. So we'll be putting in the adapters which will go and bind to the MSE1 site as well as EcoR1 site. Now, why are we doing this? Basically, adapters ligation of adapters has two functions. One is it makes the ends blunt, right? It makes the ends blunt. Second is it modifies the restriction sites. How? We'll see in the next slide. So this is the diagram for that. You have your restriction enzymes that have cut over here. You can see the sticky end over here, right? Now you're going to add the adapters. So the adapters will have a sticky end too, and it is complementary to this. For example, if it is cutting between TTAA, this is the overhanging or sticking end in your cDNA fragment. So the MS-even adapter will have A and T complementary to this. So this will come and bind over here. Same way, if this is the sticky end TTAA, that EcoR1 adapter will have a complementary AATT. So that will go and ligate over here with the help of DNA ligase. But please observe over here one small difference. Okay, what is the difference? This adapter, no, over this end, the last, we are designing it, right? So we design it in such a way that the sticky ends are matching, but the one previous to that, this nucleotide, this is G, and it has a complementary C over here. And so what will happen once this adapter goes and joins over here? Can you see what this site becomes? This becomes GTAA. But our MS-even cut site or recognition site is TTAA. But what happened? This site got modified. So no longer will MS-even enzyme be able to cut this. So adapter, after ligation, it has made the sticky end into blunt end, plus it has modified the restriction site. I hope that is clear. Same way, if you look at EcoR1 adapter, it has AATT complementary to your EcoR1 recognition site, but it has a G over here, which has a complementary C over here. So when that comes in at, instead of A over here, it is having a G, right? So your site becomes GAATTG, whereas actually EcoR1 needs GAATTC. So that is also modified. So that is the function of adapter. It makes your ends, sticky ends blunt, plus it modifies your recognition site or restriction site, so that further MS-even and EcoR1 will not be able to come and cut over here. Okay, so it stops that process over here. Or else what will happen again and again, the same procedure will have to be repeated. That puts an end to your restriction enzymes coming and cutting. Okay. This is another diagram that I have put in. Same thing, it describes the same thing. Okay, you have your adapter coming and joining. Only difference is the base is changed over here. What is the fourth step? So the first step was reverse transcription of mRNA to cDNA. Second step was restriction enzyme digestion. Third step was adapter ligation. Now the fourth step is pre-selective PCR amplification. So I told you, we'll be doing pre-selective amplification as well as selective amplification. So it's basically a PCR technique, right? cDNA-AFLP is a PCR technique. What is pre-selective PCR amplification? Let's see that. Okay. So pre-selective PCR amplification of a subset of the restriction fragments, we're going to do that. So in the first round of PCR, so if you know the basics of PCR, just to give you a brief about it, PCR basically consists of three major steps, right? The first step is denaturation, second step is annealing, third step is extension. During denaturation, your double-stranded DNA is broken down into single strands. In the second step is the annealing, where your primers come and join to these single strands. And third step is extension. So your single strands with the primer join will be, that primer will be extended so that you have a complete double-stranded DNA. So from one strand of DNA, you get one piece of DNA, you get two pieces of DNA, the same copies of DNA. Okay, so that is PCR. And all these steps happen in different temperature, and it is carried out using a thermostable enzyme called Taq polymerase, right? Not your normal polymerase, Taq polymerase, because it has to withstand high heat of 72 to 90 degrees Celsius, right? So that is PCR. So here in pre-selective PCR amplification, in the first round, pre-selective amplification is achieved with primers that complement the EcoR1 and MS-even adapters plus one additional nucleotide. So you have your cDNA fragments which are having adapters at both ends now. right? So in the first step, denaturation will happen. So you will have a single strand of cDNA with adapters at both ends. So in pre-selective amplification, you're going to use primers which will be coming and adding to the ends of these cDNA. These primers will have sequences which are complementary. Again, these primers are designed by us. So we know what is the adapter sequence. So we'll be designing it complementary to the adapter. It has sequences complementary to the adapter, plus it has another additional nucleotide. So we'll see why that is done, okay? So I'll be showing you what a primer looks like. These primers consist of three parts. It has the core or the proprietary sequence. It has enzyme specific sequence and a selective extension. I'll show you. So this is an example which shows what is the structure of primer. I hope it is clear, guys. So what is the structure of primer? You can see this is your cDNA strand, right? So when in the first step of PCR, what will happen? It will be denatured into two single strands. So when that is happening, a primer will come and join in the second step, that is the annealing step. Now this primer is designed in a particular way that it has three parts, okay? One part, the first part is the core part or the proprietary part, that is whichever company is producing these adapters now. So it will design the primer also in the same way. Adapter and primer both are designed by us, whichever company is designing, okay? So both of these will be same. So whatever is the sequence over here for the adapter, the complementary one will be over here for the primer also. So that is the core part. The second part is the complementary part for the restriction site. So this adapter has added the sticky ends of the restriction site, right? If you remember this diagram, you can see, right? So this is coming and adding over here. So this site is the restriction site, recognition site, which has been modified recognition site, okay? So it will have complementary sequence to the restriction site. Third part is the extension part. Now this could be any particular nucleotide. So here they have put C as an example, cytosine as an example. This could be A, T, C, G, depending on what kind of selection we want to do, okay? So what will happen? So for example, if you're saying that this primer is MS-E1 plus C, it will have a sequence, proprietary sequence, then the sequence for complementary to your MS-E1 recognition site plus a C at the 3' end. Same way if you're saying that EcoR1 plus A, that means it will have the proprietary sequence, then the recognition sequence for EcoR1, and at the end it will have a base A. So that is an example, okay? So this is another diagram that shows, see the last one, there will be one extra nucleotide apart from your proprietary core, core and your restriction enzyme site, and a nucleotide over here, extra one, okay? So that is your pre-selective primer. Now what is the use of this, we will see. So what happens in this pre-selective amplification? So pre-amplification with unlabeled primers having a single selective nucleotide. So what will happen? Because of this single selective nucleotides, only those fragments which has complementary G over here, right, will be attached to this primer, right? So as I told you, we're going to add restriction enzymes. We'll have numerous fragments. One end will be cut with MS-E1, one end with EcoR1, or both ends with MS-E1. So many such fragments will be there, right? This in between you will have the cDNA. On both ends you have the adapters ligated. So only such DNA fragments which has a G over here, right, will be able to bind with this particular primer. So what will happen? There itself you have kind of a selection. So if you had some, for example, you had some 1,000 cDNA fragments, it will be definitely more than that. I'm just giving you an example. If you had 1,000 cDNA fragments with adapters ligated on both ends, only like say 500 of them will have, just adjacent to the adapter, they might have a G over here. Okay, chances of that is very less. And only if there's a G will this primer come and join with this particular cDNA, right? So there you have selected out of 1,000, now 500 are only will be taken to the next step. Okay, so that is the pre-selection. It's just a very vague example, but that is how pre-selection is done. Okay, so what will happen? After this, all these will be amplified. Okay, and then you go to the next step. Next step, what is going to happen is you're going to do the actual selective PCR. So here you're going to reduce the number to say from 500 to 50 maybe. Okay, how are you going to do that? So selective PCR amplification, it reduces the further fragment number. So in the second round of PCR, so the first round of PCR is over, you have got a certain number of fragments that have been amplified. Okay, in the second round, selective amplification is achieved using selective primer. So each selective primer has an identical sequence to its corresponding pre-selective primer with an additional 2 bases. So you will have your core part, you will have your enzyme specific part, that is restriction enzyme specific part. You will also have that particular single nucleotide. Additional to that, you will have 2 more bases. Okay, so it becomes more selective. So only those cDNA fragments which has those 2 nucleotide bases will be amplified in the next step. So you can imagine the selection is, the fragment number is being reduced so much, right? So this is how it looks like. You have MS-E1 plus CGA or EcoR1 plus CAC, and you have over here MS-E1 plus CGA. Okay, so you have 3 nucleotides now. So only those cDNA fragments which has complementary T and G over here will be attached to this primer in the next PCR step. Okay, rest of the cDNA will not be amplified and will not be able to carry out the gel electrophoresis. If you know about gel electrophoresis, those bands that we visually see, they're not single pieces of DNA, right? Multiple pieces of DNA of the same size accumulate as one band, and only then we are able to see it. And that's why amplification over here is necessary. If there's just one DNA molecule, we can't visualize it. We need multiple copies, right? So now only these pieces which are having the complementary sequences to this particular primer will be able to anneal this primer to itself, and only those will be amplified. Okay, there multiple copies of those fragments will be made. Okay, and only those will be seen or visualized in the gel electrophoresis chamber. Okay, so that is what is shown over here. You have your MS-E1 primer, which is a selective primer. You have GTA over here. Okay, an EcoR1 primer with a AGC over here. So it depends which kind of primer you're going to use. That is according to our design. So this is another diagram which shows the selective PCR. Okay, so you can see that only a subset of the entire cDNA that has been fragmented will be actually going to the last step of PCR, right? Because only few of them will be having a genome sequence which is complementary to the one that is present in this primer. Only to those these primers will go and anneal, and only primer anneals, the next PCR extension reaction can happen. Okay. So what is the 6th step? So after selective PCR, now you have multiple copies of few fragments of cDNA. Okay, not the whole cDNA. You have reduced the fragments number, and you have amplified whatever fragments you have. Okay. So next step will be electrophoretic separation. So these fragments will be of different sizes. You will be carrying out electrophoretic separation. So if you know the basis of electrophoretic separation, in gel electrophoresis, you have DNA which is loaded in a well made basically in a polyacrylamide gel, right? And your DNA is negatively charged, so it will move from the negative point to the positively charged point. Okay, and in the process of that movement, the different fragments present in your sample will be separated out according to their size. The lighter ones will move faster, the heavier ones will move a bit slower. Okay, so that is gel electrophoresis. So we are going to do the electrophoretic separation of amplified DNA fragments. So labeled DNA fragments are separated by size and molecular charge by capillary electrophoresis, and the fluorescent barcode is captured by charge coupled device camera and analyzed using fragment size and genotyping software. Okay, so MS-E1 fragments are excluded from the autorad only because because only EcoR1 directed primers are normally labeled. So I had told you in the beginning, right, those fragments which have been cut by MS-E1 on both sides, we are going to avoid them during the electrophoretic separation, because there will be numerous pieces which are labeled or cut by MS-even on both sides because it's a frequent cutter. And if we take all those fragments, that is going to crowd up our gel electrophoresis. So to avoid that, your eco-carbon primer is labeled, so that is easily detected. Okay, so you can see it is detected by a charge coupled camera. So only those fragments are detected and we can analyze those. So for example, you will have so many fragments, right? cDNA fragments, and majority of them will have MS-even cuts on both ends. So to reduce the sample size, the one that we are going to analyze, we'll be labeling the eco-carbon. So those pieces will be lesser in number, right? So it could be maybe they might be cut at eco-carbon on both sides or only on one side. But out of say so many of pieces, now you need to analyze only 1, 2, 3, 4, 5 pieces. So that is how you're reducing the fragment size. Okay, the analysis becomes easier. So the bands that I showed you over here, this is the profile, right? So what and all does it depend on? Basically, it depends on the genome size. So the larger the genome size, the more cDNA you will have. And of course, even after all the steps, the number of fragments will be more for a bigger genome size, right? Second thing, it depends on how well you have designed your primer, how many selective nucleotides you have kept in your primer. So that also will depend how many fragments you get at the end, right? And also, if you have diluted your PCR product, it will be giving you those noise, or the noise will be more, the low peaks will also get magnified. So all these things affect your AFLP profile. So what does an ideal AFLP profile look like and what should be there? Okay, so an ideal AFLP profile should have well separated peaks. It should have the right number of peaks. It should have little noise, and peaks should be distributed across size range, and there should be high level of polymorphism. So this is an example that you can see over here. This is a very fine example. You can see the peaks are well separated and the size distribution is also good. But here you can see too many peaks. So if you see something like this, it's difficult to compare, right? So the next step, after you carry out the gel electrophoresis, what do we do with that? So the next step is scoring and interpretation of data. You need to do something with the data, right? So how are you going to analyze it? So typically the autorad has 100 to 300 fingerprints with sizes ranging from 80 to 500 nucleotides. So that is the size range, okay? So a genotyping software, for example, Genome Lab, automatically scores the presence or absence of amplified fragment length polymorphism generated fragments. So the differences in fragments, okay, will be scored by any software, for example, Genome Lab. This is an example of that. So electropherograms, these are called electropherograms, okay, show the AFLP profiles generated in 2 different samples by a single selective primer combination. So there is only a single selective primer over here. So each fluorescence peak corresponds to a DNA fragment with a definite size in base pair. So you can see this is one sample, this is the other sample. The difference between the 2 samples evaluated as the presence or absence of a peak is visible and constitutes the polymorphic signal that may be used for the subsequent analysis. So you have 2 different samples. Both of them have been treated with the same restriction enzymes. Both of them have been given the same primers. Both of them have been, PCR amplified in the same way. Then what you will see is if there is a difference between these 2 cDNAs, definitely that will show up over here in the electropherograms, because those differences are because of the polymorphisms. Polymorphisms could be single nucleotide polymorphism, which could have modified the bases. If the bases have been modified, recognition sites will be modified. So even if you're adding the same eco-carbon and MS-even to both the samples, because in one the polymorphism has occurred, the recognition site is modified. So if here you have a particular cut made by eco-carbon, the same cut may not be made over here in the other sample. So that particular fragment or that particular piece could be missing in the second one. So you will find out the differences between the peaks. So if here you can see clearly, right? Here these peaks are missing over here. That means those particular fragments of DNA have not been amplified. Okay, so that is how we compare between 2 samples. So that is basically cDNA-AFLP. The basic procedure we discussed was for AFLP. Only thing, in the first step, we need cDNA. Instead of doing it for a normal DNA molecule, we are going to analyze DNA and cDNA, which will actually give us the difference in gene expression between two samples. Because when we talk about cDNA, we are talking about gene expression. We are talking about mRNA levels. We are talking about transcript profiling. Okay, so I hope the steps are clear. And if you have any doubts regarding it, please do ask me in the comment section. Okay. So the AFLP technique is a powerful method for revealing genetic diversity. It was developed by Voss et al. For the analysis of plant DNA, and it has successfully been applied to several other organisms belonging to different kingdoms such as bacteria and animals. With that, we come to the end of today's session. I hope you understood what is cDNA-AFLP. If you have any doubts, please do message me in the comment box. And as always, I'll leave you with a motivational quote. The one for today is, life is inherently risky. There is only one big risk you should avoid at all costs, and that is the risk of doing nothing. It's clear, it's crystal clear, right? We cannot be saved by doing nothing. Okay, life is risky. Whatever, whether you're sitting inside a home, it is risky. So the only biggest risk that you can actually take, and which should be avoided, is not doing anything. It's the risk of not doing anything. Please take a step towards what you have been wanting to do, what your mind and heart, everything has been prompting you to do, what you have been procrastinating for so long. Take a leap of faith. Take a leap of faith and just take the first step, and things will become easier. Okay, with that I'll end. If you like this video, please do like, share, subscribe to my channel. And if you have any particular topics which you would want me to discuss, make it into a simplified, same PowerPoint presentation, please do let me know. I'll try to do justice to it as much as I can. With that, I'll leave you until next time. Bye.